Have you ever wondered how a single woman's cells changed the course of medical history forever? Today, we're diving into one of the most extraordinary true stories of our time. A tale that bridges science, ethics, and humanity in ways that will absolutely blow your mind. This isn't just another book summary. This is the story of how an African-American woman named Henrietta Lacks unknowingly became the foundation of modern medicine, and how her family fought for decades to understand what had happened to her. Rebecca Skloot's groundbreaking work represents 10 years of meticulous research, weaving together three distinct narratives that create an unforgettable tapestry of science, history, and human dignity. This isn't simply a biography or a science textbook, it's a masterful exploration of how one woman's cells became the most important tool in medicine, leading to countless medical breakthroughs, while her family remained in poverty, unaware of their mother's incredible contribution to humanity. The book follows Skloot's journey as she attempts to tell Henrietta's story, navigating the complex world of scientific research while building trust with the Lacks family. What makes this work particularly compelling is how Skloot balances the technical aspects of cellular biology with deeply personal stories of a family grappling with loss, exploitation, and the search for truth. Henrietta Lacks died in 1951 from cervical cancer, but her cells, taken without her knowledge or consent, became the first immortal human cell line. These cells, known as HeLa, have been used in research that led to the polio vaccine, cancer treatments, AIDS research, and countless other medical advances. The scientific community built billion-dollar industries around these cells, yet the Lacks family struggled financially and emotionally with their mother's legacy. This book challenges readers to confront uncomfortable questions about medical ethics, racial inequality, and the human cost of scientific progress, making it essential reading for anyone interested in the intersection of science and social justice. The narrative unfolds across three interconnected storylines that Skloot masterfully weaves together throughout the book. The first follows Henrietta Lacks herself, a young African-American woman living in Baltimore in the 1950s who sought treatment for cervical cancer at Johns Hopkins Hospital. During her treatment, doctors removed samples of her cancerous and healthy cells without her knowledge, a common practice at the time that raises profound ethical questions about informed consent and patient rights. The second storyline explores the revolutionary scientific discoveries made possible by Henrietta's cells. Unlike normal human cells that die after a few divisions, HeLa cells reproduce indefinitely in laboratory conditions, making them invaluable for research. Scientists could finally conduct experiments on living human cells, leading to breakthrough discoveries in virology, cancer research, and drug development. The cells were shipped around the world, becoming the foundation for entire research careers and pharmaceutical companies. The third and perhaps most emotionally powerful storyline follows the Lacks family's decades-long journey to understand what happened to their mother. For years, they received confusing phone calls from researchers asking about their mother's health history, not understanding that her cells were still alive and being used in laboratories worldwide. The family struggled with poverty, lack of health care, and the emotional burden of learning that their mother had achieved a kind of immortality they couldn't comprehend. Skloot spent years gaining the trust of Henrietta's daughter Deborah, who became her guide into the family's world. Through Deborah's eyes, we see the profound impact of scientific exploitation on a family already marginalized by poverty and systemic racism. The book reveals how the medical establishment's failure to communicate with the family created decades of confusion, mistrust, and pain, while highlighting the resilience and dignity of the Lacks family in their pursuit of truth and recognition for their mother's contribution to science. The story begins in 1951 when 31-year-old Henrietta Lacks visited Johns Hopkins Hospital in Baltimore, complaining of vaginal bleeding. As one of the few hospitals that treated African-American patients, Johns Hopkins was her only option for medical care. During her examination, doctors discovered a malignant tumour on her cervix and began radiation treatment, the standard care at the time. What Henrietta didn't know was that Dr. George Gay, a researcher at Johns Hopkins, had been attempting for decades to grow human cells in laboratory conditions. During Henrietta's treatment, samples of her cancerous and healthy cervical cells were taken to Gay's lab without her knowledge or consent. This practice was standard at the time, as informed consent wasn't required for tissue samples, and patients, especially African-American patients, had little power to question medical procedures. The cells from Henrietta's cervix proved extraordinary. While normal human cells died after a few divisions in laboratory conditions, her cells continued to reproduce indefinitely. Gay realized he had achieved something scientists had been trying to accomplish for years: the first immortal human cell line. He named the cells HeLa, using the first two letters of Henrietta's first and last names, though her identity remained unknown to the scientific community for decades. Meanwhile, Henrietta's condition deteriorated rapidly. The radiation treatment was unsuccessful, and the cancer spread throughout her body. She died on October 4th, 1951, at age 31, leaving behind her husband, David, and 5 children. Her family was devastated by the loss, struggling with poverty, and the challenges of raising children without their mother. They had no idea that cells from Henrietta's body continue to live and multiply in laboratories around the world. Dr. Gay began sharing HeLa cells with researchers globally, revolutionizing medical research. The cells were used to develop the polio vaccine, leading to one of the greatest public health victories of the 20th century. Jonas Salk used HeLa cells to test his vaccine, and the cells were crucial in mass-producing the vaccine that virtually eliminated polio in the developed world. This was just the beginning of HeLa's contribution to medicine. As demand for HeLa cells grew, they became the foundation of a multi-billion-dollar industry. Companies began mass-producing the cells for research, leading to countless medical breakthroughs. Scientists used HeLa cells to study cancer, AIDS, the effects of radiation, genetic research, and drug development. The cells were sent to space to study the effects of zero gravity, used in nuclear testing, and became essential tools in developing treatments for haemophilia, leukemia, and influenza. The Lacks family remained completely unaware of their mother's contribution to science for over 20 years. In 1973, researchers contacted family members asking for blood samples to study the genetics of HeLa cells. The family was confused and frightened by these requests, not understanding what scientists meant when they said Henrietta's cells were still alive. The medical establishment's failure to communicate clearly with the family created decades of confusion and mistrust. The situation became more complex when the family learned that companies were profiting from Henrietta's cells while they couldn't afford health insurance. This discovery highlighted profound inequalities in the medical system and raised questions about who should benefit from medical discoveries. The family felt exploited and angry that their mother's cells had made others wealthy while they struggled financially, unable to access the medical advances her cells had made possible. The first crucial lesson from this book concerns the fundamental importance of informed consent in medical research. Henrietta's story illustrates how the medical establishment historically treated patients, particularly minorities and the poor, as sources of research material without regard for their autonomy or dignity. Today's stringent informed consent requirements exist largely because of cases like Henrietta's, where patients were unknowingly contributing to research that would generate enormous profits for others. This teaches us that medical progress must be balanced with respect for patient rights and human dignity. The book also reveals the profound impact of systemic racism within the medical system. Henrietta's treatment at Johns Hopkins, while typical for the era, reflects broader patterns of medical exploitation of African Americans, from the Tuskegee syphilis experiments to forced sterilizations. The medical establishment has a troubled history with minority communities. Understanding this context helps explain why many minority communities remain suspicious of medical research and highlights the ongoing need for culturally sensitive healthcare approaches. Another significant takeaway involves the complex relationship between scientific advancement and social justice. HeLa cells enabled incredible medical breakthroughs that have saved millions of lives, yet the family that made these advances possible received no compensation or recognition for decades. This paradox forces us to consider whether the ends justify the means in scientific research and how we can ensure that the benefits of medical discoveries are shared more equitably. The book demonstrates the power of persistent journalism and storytelling in bringing important issues to light. Skloot's decade-long investigation transformed an obscure scientific story into a national conversation about medical ethics, racial inequality, and the human cost of scientific progress. Her work shows how skilled storytelling can make complex scientific and ethical issues accessible to general audiences, creating awareness that can drive policy changes and social progress. Family dynamics and the impact of scientific exploitation on multiple generations form another crucial theme. The Lacks families struggle to understand their mother's legacy illustrates how scientific discoveries can have profound emotional and psychological effects on families. Deborah's lifelong quest to understand her mother's story and the family's complex relationship with HeLa cells shows how scientific fame can be both a blessing and a burden for those left behind. The book also highlights the importance of science communication and the need for researchers to engage meaningfully with the communities affected by their work. The decades of confusion and mistrust between the Lacks family and the scientific community could have been avoided with better communication and genuine efforts to include the family in conversations about Henrietta's legacy. This teaches us that scientific research exists within human communities and must be conducted with respect for those communities. Economic inequality and access to healthcare represents another vital lesson from the book. The irony that the Lacks family couldn't afford health insurance while companies profited from their mother's cells underscores broader issues of healthcare access and the commercialisation of medical research. This situation raises important questions about how society should distribute the benefits of medical discoveries and whether families should receive compensation when their biological materials contribute to profitable research. The book further explores the concept of biological ownership and the legal complexities surrounding human tissue. Henrietta's case helped establish that individuals don't own their tissues once they're removed from their bodies, a principle that continues to influence medical research and biotechnology. Understanding these legal frameworks helps us navigate contemporary debates about genetic information, biobanking, and the rights of research participants. Finally, the story emphasises the importance of recognising the human beings behind scientific discoveries. For decades, Henrietta was known only as HeLa, a cell line rather than a person with a family, dreams and dignity. Skloot's work restored Henrietta's humanity to her scientific legacy, reminding us that behind every medical breakthrough are real people whose contributions deserve recognition and respect. The Immortal Life of Henrietta Lacks stands as a masterpiece of narrative non-fiction that successfully bridges the gap between scientific discovery and human experience. Skloot's achievement lies not just in her ability to make complex cellular biology accessible to general readers, but in her sensitive portrayal of how scientific progress intersects with issues of race, class, and family dynamics. The book serves as both a celebration of scientific achievement and a sobering reminder of the human costs that often accompany medical breakthroughs. What makes this work particularly powerful is its refusal to offer simple answers to complex ethical questions. Rather than condemning the researchers who used Henrietta's cells or idealising the Lacks family's response to their situation, Skloot presents a nuanced exploration of how different perspectives and experiences shaped this remarkable story. The book challenges readers to think critically about the relationship between scientific progress and social justice, while honouring both Henrietta's contribution to medicine and her family's struggle for recognition and understanding. The lasting impact of this book extends far beyond its immediate readership, influencing discussions about medical ethics, informed consent, and the need for more equitable approaches to scientific research. By giving voice to the Lacks family and restoring Henrietta's humanity to her scientific legacy, Skloot has created a work that will continue to shape conversations about medicine, ethics, and social justice for generations to come.